Hello everyone, it's a real pleasure to be here speaking to you. I'm talking from my office at Columbia University in the Big Apple in New York City. And I'm a professor here in the Applied Physics, Applied Math department. But I also have a joint appointment at Brookhaven National Laboratory, which is a very wonderful opportunity because they have fantastic X-ray synchrotron source there. I study the structure of materials, and I use neutrons, which is why I'm part of this course. But what I'm going to tell you about today will be studying nanostructure with neutrons, but also with X-rays and with electrons. So let me just get right into it and discuss what the issues are. So the main problem is actually a prerequisite in material science is knowing where the atoms are. So if you're a chemist and you make something, the first question you generally have is, okay, I've got a material, I've made a material, and I want to know where the atoms are, what's its structure. So that's a statement of the problem. And we want a solution to that problem. And there's a solution that we've had now for 100 years, which is the solution is to the crystal structure problem. So if you're able to make your material as a crystal, we have very powerful crystallographic tools for solving it. So the crystal structure problem goes a little bit like this. So you say, okay, the problem, here's my crystal, what's its structure? So let me propose a solution to you. Well, you give it to me because I'm the structure guy, and then I give it to my grad student, and she puts it on the X-ray machine, and she pushes the button, and the X-ray machine tells her the structure. That's pretty much how it goes. So I don't know how many of you have done structure solution in the lab. And so it can often go like that. And the machine tells her the structure, and then she tells me the structure, and then I tell you the structure, and then we all publish a paper together and get very famous. Okay, so that's all great. Why do I lay it out like this? And the way I lay it out like this is because this kind of problem here is a description of a largely solved problem. A problem's been solved. If you can give the problem to a machine and the machine tells you the answer, that's an example of a solved problem. So this is a bit of a problem for me when I give this talk because I have colleagues who are crystallographers and it's their profession, and they're very mad when I tell them that they're actually working on a solved problem. So there's actually something else that often happens. The machine gets stuck, doesn't tell you the structure. And when this happens, there's two MOs. Usually you either throw away the crystal, that's the most common one, and then the graduate student obviously has to go back and make a new crystal. Or you say, this is a very interesting crystal, and you make it the subject of your student's thesis. But in general, this crystal structure problem is a solved problem. Now, that's not to say that, that's not to trivialize the problem at all. So it's taken 100 years to get to this point. And it's actually it's almost exactly 100 years since the first structures were solved, and they were solved by the Bragg father and son team. And actually, it's pretty cool. You can go to the library of Leeds University. This is where the Bragg senior was working, William Henry Bragg. And this was back in 1912 when he was doing this stuff. And there's his notebook right there. And you can see the way that science was done back in 1913. You made notes with a pen, and you collect data points, and you wrote down numbers like this. And this is the real notebook, which resulted in their first structure solutions, which resulted in their Nobel Prize. So we can look at this page. This is actually I'm not sure if this is the exact one, but this is what goniometer looked like in those days. So people measured crystals way before they had X-rays, and what they did is they measured the angles between facets on the crystals, and from that they deduced internal symmetries. So they actually had devices that looked like this, where there were no X-rays coming in, but you put a crystal somewhere here right on the machine, and you could measure the angles between the faces. So what Bragg did is he took a machine that looked like this, and he attached it to a Coolidge tube or one of these new X-ray producing tubes. And it was the quality of the data that they were able to get that allowed them to start doing structure solutions. And here we are on page 11. This is the world's first Bragg peaks right here. They got named after the Braggs. And they're sharp peaks that came from very characteristic planar spacings in the structure. We all know this from our introductory chemistry and physics courses. And this is the first structure solution. They've already here figured out that you can express the planes in terms of HKLs here, and they're solving it, and it's I think it's sodium chloride. So they solved their first structure on page 43, and as a result of that, they won a Nobel Prize. So back in the day, it was easy. You could just have a paper notebook, and on page 43, you do something, and you win a Nobel Prize. So much harder these days. Of course, it was hard for them. They had to build all the machinery. But anyway, so that's 100 years ago, and we've come a really long way. So it's highly automated, and they're very powerful algorithms, but it's a really extremely non-trivial problem. So on the IUCR website, IUCR stands for International Union of Crystallography, they actually list all of the Nobel Prize winners that are associated with crystallography. And I have to admit that some of them are rather loosely associated with crystallography, but nonetheless, the ability to solve structure is incredibly important, and it's been a very rich source of Nobel Prizes over the years. And so this is the top of the list, and it doesn't fit on the slide, so I have to make the words smaller. So here they are in smaller letters, but it still doesn't fit on the slide here. So to fit them all on the slide, the letters have to be this small. So that's how many Nobel Prizes the IUCR thinks has been associated with crystallography. That's how important the problem is. That's how hard it's been to solve, and how many creative people have put to work and managed to solve it. Okay, so that's great. We know how to get the structure of materials, job done, right? Okay, but now we're interested in nanomaterials. There's a nano revolution going on, as we know. So actually, it's a different problem that we want to solve. It's not a crystal structure problem, it's a nanostructure problem. So the problem is very similar to the crystal structure problem. You're a chemist, you've made something, but now what you've made is a nanoparticle. But you still want to know its structure, you still want to know where the atoms are. All right, well, in science, what we tend to do is we tend to try and we stand on the shoulders of giants, as it were. So what that really means is we copy each other. So what we're going to do is we're going to try the same solution. So you're a chemist, you give me the nanoparticle. You give it to me, I give it to my grad student. She puts it on the X-ray machine and pushes a button. And what comes out? Well, actually, if you put a single nanoparticle on an X-ray machine, nothing comes out, because there's not enough scattering strength. There aren't enough X-rays coming out of your X-ray machine, and there aren't enough atoms on the nanoparticle to scatter, so you get basically nothing. But what we can do is we can take a lot of these nanoparticles, and we can make a powder of them, and we can put them into a capillary, and we can put that in the beam, and then we do get a signal out. But the machine doesn't tell us the structure. What comes out is something that looks like this. So this is actually a real diffraction pattern, X-ray diffraction pattern from cadmium selenide nanoparticle, which would look something a bit like this thing up here. And this is the pattern that comes out. And you can see it's just a series of broad bumps. It's not a structure. The structure would be a set of coordinates telling you where every atom is on that ball up there. So this is the nanostructure problem, and whereas the crystal structure problem is a solved problem, the nanostructure problem is not a solved problem. It's essentially unsolved, and this is the one that we're working on, and we're very interested in working on. So there's been actually a lot of progress in this regard over the last number of years, and as with crystal structure solution, you can take two approaches. The most common approach in crystal structure solution is what's called single crystal crystallography, where you have a single crystal and you measure the Bragg peaks in all the directions, and then you use the intensities and the positions to determine where the atoms were using some very fancy inversion algorithm and a very, very big computer. So that's single crystal crystallography. The other approach is powder crystallography, where you take your single crystal, you grind it up into a powder, and you measure the data from the powder. And the question is, why would you do that if you had a beautiful single crystal? And the answer is, of course, actually you don't do that if you have a lovely single crystal. But there are many, many cases where you don't have a lovely single crystal and you take the powder approach. But these are two successful approaches to solving the crystal structure problem. So we can kind of look at the nanostructure problem in the same way. So one approach is to do single crystal nanocrystallography. And this is not something that I do, but there's people around the country and around the world that are doing this very successfully. John Spence at Arizona State, and John Miao at UCLA, and Jim Zhou at Illinois. So a number of very great people, and there are many others. And basically the idea is that you get a single nanoparticle, you have to figure out how to mount it, you have to figure out how to get enough X-rays, neutrons, electrons onto the sample to get significant scattering, and then how to invert the problem. But there's some notable successes. And this is one example which comes from actually Jim Zhou's group. And basically what he did is he put these double-walled carbon nanotube. This is this here is a simulation of it. This is not a real image. This is a simulation of it coming from a computer. He put it in an electron microscope. He illuminated it with a very coherent beam. And then it's possible actually using when you're in a condition called an oversampled condition, there's actually sufficient information to do the inversion. And this is an actual image that he got from the diffraction pattern. So this is a diffraction pattern he got from this double-walled carbon nanotube in his electron microscope. After doing this inversion algorithm over here, he got this image here. And then this is a kind of a simulation of what this would look like from a computer. So you can actually kind of see the structure here, but this is just an image. It's not really a structure. So what we mean by a structure is it's a set of coordinates. And what we actually have here is an image, a high-resolution image with blobs. So to turn this into a structure, we need to get a three-dimensional image of this. And we need to also do some kind of tomographic reconstruction that tells you exactly where the atoms are located in 3D. So this is ongoing work, and it looks very promising. And we have very powerful sources, like modern aberration-corrected electron microscopes, and also linear coherent light source, for example, free electron lasers. So very exciting area, but no cigar. They haven't basically solved any structure that anyone wants to know the answer to. But we're getting there. The other approach, which I'm going to talk about, is taking the other approach of powder nanocrystallography, where you take hundreds and thousands of your nanoparticles and you put them into a vial, into a capillary. This is actually a capillary filled with a crystalline powder, and this is a 2D diffraction pattern from a powder of crystals. So I'm just using these for illustrative purposes. But what we want to do is we want to fill the capillary with powders of nanoparticles, and we want to collect a diffraction pattern that will look something like this, and then we want to find ways of inverting it to get the structure out. So that's the approach we're taking, and also it's a hard problem, and we're making progress, and I'll tell you a little bit about that. So let's think a little bit about why the nanostructure problem is actually so hard. So what we can do is we can think of neutron scattering, X-ray neutron scattering. This is all kinematical scattering. It takes place in the weak scattering limit. And what I've got here is I've got a simulation of two square lattices. So this was a simulation that I did with Thomas Proffen, who was a postdoc with me. He's now a big shot manager guy at Oak Ridge at the spatially neutron source. But back in the day, we were working together at Michigan State, and we did this simulation. So these are actually 3D crystals, simulated crystals, and they're a square lattice. And they have 70% of black atoms, and they have a 30% of yellow atoms. Now, if you look at the one on the left and you look at the one on the right, your eye immediately tells you that they're different. There's no problem with that. And the reason your eye is so good at that is because you've basically evolved over time, over the history of mankind, to have a very good pattern recognition algorithm in your brain. And the reason you want a good pattern recognition algorithm is you want to be able to tell the difference between your mother and a tiger. If it's your mother, obviously you reach out for a hug, and if it's a tiger, you run away. So that's how you've evolved. But what I'm going to show you is if you do a conventional powder diffraction pattern, you can't actually tell the difference between the left and the right. So these two crystals are the same in respect to the fact that they're 2D crystals with 70% black atoms and 30% of white. In the left-hand side, the yellow atoms have been placed randomly on the lattice. In the right-hand side, we've introduced some kind of tendency for clustering. So if you're a yellow atom, there's a higher than average probability that your neighbor is yellow, and likewise for black. But there's enough randomness that it doesn't order over long range. Okay, so these are two simulated crystals. Let's take a look at what the neutron powder diffraction pattern would look like from them. And it looks like this. If you have these lovely Bragg peaks, which are coming from the crystal structure, but if you look at the pattern on the left and the pattern on the right, they're identical. So there's basically no information in the Bragg scattering to tell you about whether the yellow atoms are short-range ordered or not. Now, if you look very carefully at this intensity scale, and it's a little bit hard to see, but this number is 4, and this is a 10 to the 3, I think. It's hard for me to even see. What we're going to do is we're going to blow up the scale by 1,000 times, which means that we're going to focus down on the bottom of the scale right down here. So this is what's in this next slide. So these sharp peaks here are the old Bragg peaks that you saw. And now what we've done is we've plotted the powder diffraction pattern from the one on the left in red. So this guy is in red, and this guy, the scattering from this one, is in blue. They both have the Bragg peaks. You can't see the blue ones because the red ones are plotted on top, but the blue ones have Bragg peaks too. But what you see is that the random case, there's a monotonically decreasing diffuse scattering signal. So it's like the Bragg peaks sit on a sloping background down here. But in the case where we have short-range order, nanoscale order, if these were atoms, we have some kind of structured diffuse scattering down here. And so it's the peaks in this diffuse scattering which are containing the information about whether the yellow atoms are short-range ordered or not. And it's a statement of the fact that nanoscale order is contained in the diffuse component of the scattering in a diffraction experiment. So we're still going to do diffraction experiments to try and solve the structure of these nanoparticles, but we're going to not concentrate on Bragg peaks, we're going to concentrate on the diffuse scattering part. And then we have a problem of signal to noise, because you can see that even though these data were simulated, we didn't have enough statistics on our simulation, and so they appear noisy. And when you do real experiments, the diffuse scattering also has to be measured with very good statistics. Now there's an information theoretic question that you can ask, which is that if you measure a diffraction pattern and all it has in it is 4 peaks, if you look at this blue one, it just has 4 peaks in it. Each peak contains about 3 bits of information if it's a Gaussian. It's the position, the width, and the integrated intensity. So we have 4 of them, so we have 12 bits of information. So 12 bits of information will allow us to locate about 4 atoms. But maybe the cluster has a larger number of atoms. It seems like there's not enough information for us to actually solve this problem. On the other hand, if you look at the Bragg scattering, this is actually Bragg scattering from some pharmaceutical compounds. And basically, we have a small molecule system here which forms into 2 different polymorphs, a beta phase and a gamma phase. So they're the same molecules, but they're forming into different crystal structures. And they give rise to these different patterns of Bragg peaks. And you can see that these are very, very different. And what the people in the drug industry do is they use this for fingerprinting. So what you can do is you can take any pill off the shelf in the chemist, in the pharmacy, and you can grind it up and you can put it on an X-ray machine, and you'll get Bragg peaks like this. And the Bragg peaks will tell you what the crystal structures of the, it won't tell you the structure, but it'll give you this kind of fingerprint of all the constituents in that pill. And what the drug companies want to do for quality control is they want to make sure that what's in the pill is what they think is in the pill, because you're going to take that pill to make you better, and the drug company wants the pill to make you better and not make you sicker. So it's very important to them that they know that what they think is in the pill, and they need this way of fingerprinting. And what they do is they have these very rich patterns of Bragg peaks to do this. But let's say that you had a drug which was in either amorphous form, so completely disordered molecules, or nanocrystalline form. So it looked like a nanocrystal, like that cadmium selenide I showed you. What happens then? And what happens then is if you put it on the same powder diffraction machine, you get out this diffuse scattering. And so this thing looks very like the pattern that we got from the short-range ordered yellow atoms. We basically have 1, 2, 3 peaks, and here's a shoulder. So we have, again, about 12 bits of information. But it's very difficult for the drug companies to use this for fingerprinting. So this is something that I'm going to come back to later. But you might argue that when you have these nanoparticles, you're never going to solve the structure because you always have very limited information content in the diffuse scattering data. So one of our objectives is to get around this problem. And it turns out that we can do that by actually measuring to high Q. So let's go back to this cadmium selenide nanoparticle example. And remember that you made this cadmium selenide nanoparticle over here, and then you wanted to know its structure, so you gave it to me, and I gave it to my student, and my student put it on the machine, and this came out. And this is immediately looking like this diffuse scattering pattern that we got from the yellow atoms again. It's just got a few broad peaks. And then you see there's a very long tail out here where there's nothing. And so it looks hopeless that we're just left with this very small signal here. But it turns out that what I'm going to tell you about a little later is a pair distribution function method for analyzing data like this. That actually we analyze a function which is not the raw intensity, which is shown over here, but it's actually this little function q times s of q minus 1, and s of q is this scattering intensity over here. But it's been normalized by dividing by the atomic form factor. So the atomic form factor in X-rays is something which falls off with increasing distance. So it looks like a smooth curve falling off like this. And we're going to divide the blue curve by the red curve. And when we do this, we get something that looks like this. So now actually what's happening, if you look over here, this number here is 20 inverse angstroms, 24 inverse angstroms. If we look over here, there's actually a very strong signal out in this high Q region. So Q is the momentum transfer of the measurement, which is related to the angle. So Q is 2 theta sine, is 4 pi sine theta over lambda. So it depends on the scattering angle, and it depends inversely on the wavelength of the particle. So the bottom line is that we can get much more information if we measure data to these very high Qs, which means we use short wavelength probe, or it means that we measure over a wide angular range. So we're going to try and solve the nanostructure problem by measuring the diffuse scattering, because that's where the signal is, but measuring it over a wide Q range. So I'd just like to summarize this introductory part of the talk here. We can easily collect data from powder samples of nanoparticles. And when I say nanoparticles, we can also look at molecular systems. We can look at molecular systems in solution. We can look at colloidal suspensions. We can look at all of these kind of things. But where the objects themselves are nanoscale, we can easily collect data from this kind of, there's not a problem collecting data. We don't have to isolate a single nanoparticle. We can just look at the powder in a capillary. On the other hand, there's a very big loss of information when we do this. The scattering data appears in a broad diffuse scattering signal. The information content seems to be limited in quantity. But by doing rather special experiments where we go to high momentum transfer, which means high 2 theta, and using short wavelength probes, we can actually uncover much more information. And it turns out that we can we can make a lot of progress in a lot of interesting systems. Okay, so that's an introduction to the whole thing. I wonder if anyone has any questions at this point, because I'm going to shift gear a little bit and talk about some of the scientific results that we can get with this approach. Actually, before I get to the science, I'm going to tell a little bit about the technique. So the Braggs got the Nobel Prize for discovering Bragg scattering, which was named after them. And here's the world's first Bragg peak. I already showed it to you, and it was on page 11 of their notebook. But actually, it's a little known fact that the Braggs actually discovered diffuse scattering, and they discovered it before they discovered Bragg scattering. So the Braggs discovered diffuse scattering on page 11. So remember, they discovered Bragg scattering on page 11, but they discovered diffuse scattering. So here was one of their even earlier measurements where they couldn't resolve a Bragg peak. This is basically diffuse scattering, and they discovered it on a, what is it said? Oh, it's not said. Oh, page 5. So they discovered diffuse scattering on page 5. So it's a complete misnomer. Basically, the Braggs discovered diffuse scattering first. So in my opinion, in my personal opinion, we should call diffuse scattering as Bragg scattering and name those sharp peaks after something else. Okay, anyway, so I want to say a little bit about this atomic pair distribution function, or PDF. And this has grown in popularity over the last 15 years or so because we've actually managed to develop the experiments and the methodology for analyzing the data to the point that it's actually proving to be very useful in a wide range of systems. So we're basically studying the diffuse scattering, which is what I told you. So what we do is we take our sample, which is a powder sample, we put it in a beam, and we collect data over a wide angular range. So we can put detectors all around the sample here, and we collect data over a wide angular range. And what we get is something, if we have a crystalline material, we get something that looks like a Bragg pattern, but there's also a diffuse component in here in general. And if it's not crystalline at all, there's no Bragg peaks, but there's still a diffuse part. But what I told you at great length before is that we measure the data over a very wide Q range. So this is a Q of 30 inverse angstroms. Now, if you try to do this experiment on a copper K alpha X-ray source, which is a common source in labs, or if you try to do it on a usual neutron powder diffractometer, a normal one at a reactor source, you would only get the Bragg peaks that are very low down here in the low Q region. So you'd only get these guys. Because we're collecting over a very wide Q range, we get many Bragg peaks, but we also get diffuse scattering up at high Q. And what we do is we take the data, we divide by the atomic form factor, we normalize it, and we multiply by Q to get this kernel. And you can see that this is actually a sine Fourier transform. We have a kernel here, Q S of Q minus 1, and then we have sine Q R dq. Sine Q R dq, yeah, okay, and 2 pi and 2 over pi, and we get G of R. So what we're doing is we're sampling the data with sine waves, and then we're integrating those sample data, and we get this G of R. So G of R is a function in real space. It's a little small here to see. But on this axis, this axis is R, and this unit is angstroms, and this number here is 10. So 10 angstroms is 1 nanometer. So right here is 1 nanometer. So this point right here is 1 nanometer. This axis here is G of R. G of R is a probability. It's a probability of finding 2 atoms separated by this distance over here. So what you can see is that there's actually a high probability of finding, in this particular material, there's a high probability of finding 2 atoms separated by a distance a little bit less than 1 nanometer. There's a very low probability of finding atoms separated by this distance. And of course, there's a 0 probability of finding atoms closer than the nearest neighbor distance. And then this is a negative peak, which is a slight complication because these were neutron data, and it's got an atom in which has a negative scattering length. So this negative peak is actually an inverted positive peak. But there's actually a high probability of finding atoms separated. Let's move to this one. High probability of finding atoms separated at this distance. And this is because of the well-defined structure in this material. It turns out it's a perovskite. They have octahedra of atoms, and this is the oxygen-oxygen distance on the octahedra. But we have this function which has sharp peaks, and the peaks basically give us the distances that separate atoms in the solid. And we can use this actually for solving the structure. So we can get an intuitive field with C60. So C60 is familiar to most people. So we got a sample of C60, and the C60 is a molecule with 60 atoms, and the molecule forms into an FCC solid. And what we did is we put this in a capillary and we measured it, and we got the diffuse scattering, and diffuse scattering looked like this. And then we did the Fourier transform, we did data corrections in the Fourier transform, we got the PDF, which looks like this. And the first peak in the PDF is right here. This is 1.4 angstroms, and this is the carbon-to-carbon nearest neighbor distance in the material. The second distance here is the second neighbor distance, which is also carbon-to-carbon, but across the hexagon here. And the third neighbor distance is this one here. And this is the atoms that are diametrically across a hexagon. It's maybe a little hard to see, but it's these guys. Now, the C60 is made up of hexagons and pentagons. So here's a hexagon, 1, 2, 3, 4, 5, 6. So that's that hexagon. Here's a pentagon up here, 1, 2, 3, 4, 5. And if you look at the pentagon, it has the near neighbor distance. It has, so these two are separated by the near neighbor distance. And it has the second neighbor distance. These two are separated by the second neighbor distance. But there's no third neighbor distance on the pentagon. So immediately, intuitively, you can see if we look at the relative intensity of this third peak to this second peak, it actually tells you the relative number of pentagons and hexagons in the structure. If there were more hexagons, this peak would be stronger. And if there are more pentagons, this peak would be weaker with respect to that one. So this helps you intuitively to start to build up a feeling for the PDF. We also have, here's an atom here, and the atom on the molecule that's the farthest distance away is the one that's diametrically opposite, is that one. So the longest intraatomic distance on the molecule is basically the diameter of the molecule, and that's actually 7.1 angstroms. And if we look in the PDF, and remember this PDF was just a Fourier transform of this diffuse scattering, and we see the last sharp peak in the PDF, this is a 6 and this is an 8. So this last sharp peak is exactly at 7, actually 7.1. And so this PDF here, which is coming just as a Fourier transform of diffuse scattering, it has all the positions of the atoms in the C60, and we've later shown that we can actually solve the structure of C60 from this information here. Now, if you look carefully, there are some broad peaks also out here. And these are actually coming from the intermolecular correlation function. They're how the balls pack. So what's happening here is I have a ball that's here, and I have a neighboring ball that's there. And it turns out that the ball-ball separation, the inter-ball separation, is 10 angstroms. And so the first broad peak is actually peaked at 10 angstroms or 1 nanometer. And the balls form into an FCC structure. So the second neighbor distance is root 2 times 10, and the third neighbor distance is root 3 times 10. So this is 10. Root 2 times 10 is a little over 14. Root 3 times 10 is a little over 17. And so these peaks here are actually giving information about the packing of the molecules. So if you have a molecular system, in principle, if you're lucky and if it's a rigid system and has high enough symmetry that we can actually solve it, you can get intramolecular, you can get the structure of the molecule itself from this region of the PDF, but you can also get information about the packing of the molecules from this higher R region. And there's a very nice separation in the PDF between the low R and the high R signal that helps us to do this separation. So this is giving some justification for why this approach is actually very useful when you want to solve structure of a nanoparticle or a nanosystem. Now we're able to do the forward problem very simple. What's the forward problem? The forward problem is given this structure here, what's the PDF? All that means is that we need an algorithm that can take the atomic coordinates and calculate the PDF. And that's an analytic, essentially, well, it's normally done numerically, but it's a very straightforward problem to solve. The inverse problem is much harder. The inverse problem is, here's a PDF. What's the structure that gave rise to it? And we only have a few examples where we've actually solved the inverse problem from the PDF to get the structure of a material, but it has been done. But the forward problem is very trivial, and this is the one that we use the most actually in our modeling. So basically, the way that we calculate the PDF from a structure is, let's do this one, which is graphene. We pick an atom and we put it at the origin. And then what we do is we go out some distance in space until we find another atom. So actually what we do is we draw a circle around, or a sphere around, this central atom and it turns out that at 1.4 angstroms away from this central atom, there's another atom, and there's actually 3 of them. So what we do is we put a peak in our PDF at 1.42 angstroms, and the height of that peak depends on the scattering power of these guys, and it depends on the number of these guys. And then we just keep going. So then we go a little bit further out until we find another coordination shell. So here's another coordination shell. This one has twice as many atoms in it. There are 6, and it's this guy here. And then we keep going a little further, and here's the next coordination shell, which is turns out to be very close. So there's another atom, but another peak in the PDF. But this one's weaker because it has fewer atoms in this shell. So you can kind of see how the structure gets built up. And we can just keep going out, putting more circles, and then putting more peaks in our PDF. And you can see the PDF gets built up like this. And we have programs for doing this now, where we can do regression. So what we can do is we can calculate the PDF from a model, we can compare it to some measured data, and then we can update model parameters in such a way as to get a good fit to the data. So the blue curve here is, these are real measured data coming from nickel, which is FCC metal. It's a crystalline material. And the red curve is the calculated PDF from the structure of nickel, the FCC structure of nickel. And you can see that we get very good agreement. This is the difference curve below, and you can see we get, apart from some noise ripples, we get very good agreement. So when there's a well-defined structure, we can calculate it, and we can extract quantitative structural parameters by fitting the data. And so this is what we do a lot of. So the general strategy for studying nanostructure then is we collect data to high Q with good statistics. We need short wavelength probes. We make corrections to the data to extract the coherent scattering from all the backgrounds and all of the other stuff going on in there, any kind of incoherent scattering. We remove. You can analyze the data in Q space, but we like to actually Fourier transform it into real space to get the PDF. And then we create models to simulate the data. So this is the basic strategy. All right, so again, let me give an opportunity if you want to ask a question. I'll just pause for a second. But what we're going to do is we're going to take this method and see what scientific things we can learn. Okay, so I'm going to have to go a little bit quickly through this. I have more examples than I really have time to talk about, but you can see my view graphs after the talk. And there's also some other resources on the slides that you can go back and look at. But this is just giving you a taste of what you can learn by using this kind of approach. So the one example was the one I started with, which was the cadmium selenide quantum dot. So these are interesting little materials where you just take a semiconducting material and you make it so small that you get quantum confinement effects, and then you get modified properties. And these are of interest, for example, in the light collecting layer of solar cells, and they're also used as dyes, and somebody's even actually commercializing quantum dots. Yep, they're that small. This is an artist's impression of what they look like. Everything is very ordered and lovely in here, and there's a nice capping of organic ligands. So in order to stabilize these small objects, you have to cover them in surfactant organics. So this is them inside the solar cell, and then actually they have very good optical properties. This is a photoluminescence, and in each of these vials are the same material, cadmium selenide, but different sizes, and that changes the band gap, which causes them to photoluminesce with a different color. So these are also of interest for ambient lighting. So what we do then is we collect the data, and of course we get diffuse scattering patterns, as I've discussed, and we Fourier transform and we get PDFs. Now this is the PDF of a bulk cadmium selenide semiconductor, and these are the quantum dots. And what you can see is that the local structure is very similar. We have peaks always in the same positions, so the local structure looks like the bulk. And there's two competing structures for these. One is the wurtzite, which is a rhombohedral structure, and the other is zinc blende, which is a cubic structure. So what we've done here is we've taken the blue curves are always the measured PDFs from the bulk and from nanoparticles, large, medium, and small. And the red curve is actually a fitted model. And if you look at the bulk material, the wurtzite, this is the difference curve, so the wurtzite fits much better to the bulk than zinc blende. So the bulk material is the wurtzite rhombohedral structure. When you look at the nanoparticles, you can see that the fit is actually quite good or quite bad, depending how you want to look at it, for the two different structures. So what's going on? Well, the main difference between the zinc blende and the wurtzite structure is they have the same local geometry, which is a corner-shared tetrahedral geometry, but they have different packing. So the wurtzite structure has A, B packing, stacking along a certain crystallographic direction, and along the same kind of structural direction, the zinc blende has A, B, C stacking. So it's just a stacking difference. Now, another big difference is that if you look at the bulk PDF, you see peaks extending out to high R. This is R. In the nanoparticles, the intensity of the peaks falls off. There's an envelope killing the structure. And if we assume it's a spherical particles, we know what the functional form for that envelope is. It looks like this. And so we can actually take our structural model for the bulk material, and we can attenuate. So we'll calculate the PDF for the bulk, and we'll attenuate it with this model, and we'll give it one parameter, which is d, the diameter of the balls. And this is the kind of fit that we can get here. And you can see that the fit is doing very, very well, but we're calculating rather well this drop-off in intensity. And when we do that, we can actually obtain from the data what the size of the nanoparticles was. And bear in mind, this is the average size over all the sample. And we get a 3.7 angstrom nanoparticle. And other measures of size, like directly observing it in TEM or using some calibration curve of the UV vis absorption or the photoluminescence, we're in good agreement. We're much more sensitive to the size when we have than other techniques when we have a very small nanoparticles. So we can get the size, we can get the structure. We can also measure strain in these. You might want to know whether there's any kind of strain in the material, whether the bonds are shorter or not. So what we can do is we can look at the nearest neighbor peak, which is cadmium selenide peak. And what you see is as the nanoparticles get smaller, this peak shifts to the left. In other words, the cadmium-selenium bond is getting shorter as the size of the nanoparticles gets smaller. And we can express that as a bond strain, which is the change in length over the length. And you see it goes down to, there's almost a 1% strain, which is a massive strain in these very small nanoparticles. And one possibility for that is a surface tension, but it's obviously actually essentially the nanoparticle is minimizing its total energy. So the average strain, which is the position of this peak, shifts to the bonds shorten, but the width of this peak also broadens. What that means is there's an increased distribution of bond lengths in the material. So there are some shorter and some longer bond lengths in there. And we can also trace the inhomogeneous strain. So we can measure the structure, we can measure the size, we can measure the shape, we can measure the strain in these materials. So it's remarkable how much information you can get out. We can also, by doing a little trick, measure the bond strength. So what we can do is we can look at the temperature dependence of the atomic displacement parameters. So we can look at the width. We can look at the width of this peak here, and we can plot it as a function of temperature. And that tells us how soft or hard the bonds are, and then we can compare that to the nanoparticle size. So these are actually platinum nanoparticles, which are used in catalysts, but this is the bulk material, and this is the temperature dependence of the ADP, the atomic displacement parameter. And you can see that the ADP gets larger for the smaller nanoparticles. That's just also reflecting the fact that there's more inhomogeneous strain in those nanoparticles. But if you actually fit a Debye or an Einstein temperature to this, which is a measure of the stiffness of the bonding, that actually is constant until you get to the smallest nanoparticles, and then it falls off very rapidly. So the smallest nanoparticles are much softer. The bonding is less stiff. So we can extract all of these things from the PDF. So I don't know, this is a soft condensed matter course. I don't know how many of you are interested in platinum or cadmium selenide, but nonetheless, the general principles are the same, that we can apply this and extract very high quality quantitative information, even from nanoparticles. Okay, I'm going to go rapidly through this example, but there are different kinds of nanomaterials, and one of them are nanoporous materials. This is a kind of a material which is a bulk material, but it has nano-sized holes in it. And it's useful because you want to actually put nanoparticles into this porous host and have them do interesting things. So one example of this is you can take a silica material and you can make very small holes in it, and you can functionalize the surface inside this confined geometry, and then you can bind things in. And so this was a collaboration with Tom Pinavaya at Michigan State University, and he's actually got some thiol functionalized silica, and it's very good at binding mercury. And this is what the mercaptan molecule is that's functionalizing the nanopores, and this is the thing that binds the mercury so well. And we collected data, and the data didn't look like much. They had a very strong silica signal, and there's just a few peaks that you can see. But we see that this is the pure silica right here, and then when we load the silica up with mercury, we see some peaks that are appearing. So we can do a nice difference method, where we subtract away the silica host, and we're just left with the signal coming from the intercalated compound. And then we can just look at where the peaks are in this. And what we did here is we compared it to some analog crystal structures. So this is cinnabar, this is a mercury oxide. And just by looking at where the peaks were in these compounds, we could line them up with our test compounds. So these are our test materials. So what this shows is that this is the peak. This is where the peak would be in mercury oxide, but there's no signal there in this material. So there's no mercury-oxygen binding, which is in disagreement with an earlier XF's experiment, but there is mercury-sulfur binding. In cinnabar, we see a strong peak here in our material, and in cinnabar there are two peaks coming from mercury-mercury distances, like this one. One is on the cinnabar chain, which is a one-dimensional chain of mercury and sulfur. And it turns out that we're getting these, what we could deduce from the data is that we're getting these chains of mercury-sulfur inside the pores of silica, but we're not getting any ordering between the chains. So I don't want to dwell on this one. Let me just move on. Now, let's say that you're interested in more soft materials, which I think most of you are, and you have them floating around in a solvent. Because we're doing this so-called powder measurement, and we're looking at distribution of these materials, we can actually look at these in solution. So what we were looking at here were the precursors to gold nanoparticles. So if you want to make gold nanoparticles, it's often done by a solution method, where you have these molecules that they float around in solution. They contain gold and sulfur, and it's not clear exactly what the species is that's floating around in the solvent, and it's going to give rise to the gold nanoparticles. And these were some candidates from the literature, but we can actually measure this, and we can subtract the solvent signal, and we were left with this PDF, and it shows us exactly where the peaks are on these little clusters that are in solution. And it turns out that actually this one, which is made up of four gold atoms with sulfur staples holding them together in a ring, this is actually the precursor cluster to this gold nanoparticle formation. And we could extract that by getting the signal from the solvent in solution. And recently, we've been working with some pharmaceutical companies to look at nanoparticulate drugs, and they're floating around also in suspended solvents. So this is a drug which is used in a nebulizer. It's an anti-asthma drug, and it's actually sitting in a suspension. So these are capillaries that are filled with solvent with this nanoparticulate drug in. And what we can do here is we can get the PDF of the solvent, which is shown in blue here, and we can get the PDF of the crystal structure of the API, the active pharmaceutical ingredient. And then we can look at our materials in suspension. So this is two PDFs from two different samples, and you can see the degree of reproducibility. So we definitely have a very good signal of what the API, what the drug molecule looks like when it's present. And now what we can do is we can look at more and more dilute solvents. And so we have here 5 weight% of API in the solvent. And you can see the blue curve is 5 weight% API, and the signal is in strong agreement with the crystal structure of this. But we've now gone to 5 weight% down to 0.66, down to 0.25 weight%. So these are extremely small concentrations of a low Z material, an organic nanoparticle floating around in an aqueous solvent. And you can see that the agreement between these curves is absolutely remarkable. So we're confident now that we can actually study small molecular clusters floating around in solvents and in suspension at rather low concentrations. I think this one is another pharmaceutical example where we were looking at the ability of the PDF to fingerprint. So I talked about this example earlier, and it's actually carbamazepine, which is an anti-epileptic drug, and this is what the fingerprints of the two different crystalline forms looks like. And you can turn this into an amorphous form by melting. You actually melt this gamma form here, and then you quench it very quickly, and it forms an amorphous material, and you just get the diffuse scattering like this. And if you're going to fingerprint, you want to know what the packing looks like. So you want to say, is this melt-quenched material, are the molecules packing in the gamma form, or are they packing in the beta form? So you want to look at this green curve, and you say, is it more like this one or that one? And there's no hope, basically. But if we do a total scattering PDF measurement where we measure these are the data measured over a narrow range of Q, but these are the data measured over a wide range of Q. We can start these are the data measured over a wide range of Q, and these are the data which have been Fourier transformed to the PDF. And what you can see is the low R peaks are all identical, as they must be, because the low R peaks are coming from the molecule itself. So it's this high R signal, which is coming from the molecular packing. And it looks like there's not much signal there, but what you can do is, this is the low R region, but instead of focusing on the low R region, let's expand this region here and look at it on a wider scale. And actually, this is what the signal looks like in that region. And it looks like kind of like noise, so maybe we just have noise, but actually it's not noise, this is the real signal. And this is the gamma crystalline form. And if we plot on top of it the melt-quenched form, you see it actually looks very, very similar to the gamma crystalline form. So the melt-quenched form is green, the gamma crystalline form is blue, and the only difference is that the intensity of the green peaks is falling off. And that's again because it's a nanocrystalline material. So we can correct the green data for the nanocrystallinity by dividing by this form factor curve that I talked about before. And it looks like this. And you can see there's a very good agreement between the green curve and the blue curve, telling you that actually this material is mostly actually the molecules, although it's nanocrystalline, the molecules are packing in the gamma form. So again, there's a lot of information that you can get about some molecular system. This is an example which just shows how you can do phase quantification. So we're actually mixing together an amorphous and a crystalline form, and we're extracting the quantities of each. And there's one last little view to the future that I wanted to show you, which is we can now do all of this in a spatially resolved way by combining PDF with tomography. So this is an experiment that we just did last year at the SRF in France, and it just got published in Nature Communications. And basically what we can do is we take 10,000 PDFs with the beam going through the sample. So the beam just goes through the sample like this. And we get 10,000 PDFs, and then we do a tomographic back projection, and we produce this pixelated image where every one of these dots on here is actually a pixel in the image, and we have a complete PDF in each of these pixels. And so we can, this was a catalytic sample where it had a palladium catalyst sitting on an alumina surface. And if you look at the surface of the catalyst body, it actually had palladium nanoparticles that were large. They had PDF peaks extending out to high R. If you look deep inside the material, there was less palladium in there, and the size of the nanoparticles was smaller. And we can spatially resolve the quantity and the size of the nanoparticles as a function of position. And this catalyst body was actually sitting inside a very thick-walled glass tube. And this technique was very nice too, because it allows us to separate the signal from the sample holder from the signal coming from the sample itself. And so the PDFs we have in here have no signal at all in them coming from the holder, because we've separated it tomographically, which means that we can actually now do very nice experiments in special environments that would normally be very challenging. So you could, for example, look at a material in a high pressure cell with a rather thick wall, and we can separate the signal coming from the cell. So that's everything I wanted to say. That gives some example, tells you what we're trying to do, tells you the approaches you can take. And now I'm more or less finished, except I want to just very quickly show you some resources that are available. Let's say you're interested in applying this in your research. How do you go about it? Where do you do it? So I'm just going to run through this, but basically this information will be on these view graphs, which will be available later. Getting started. Okay, think about doing a summer school, do background reading, contact someone who's doing it to collaborate for the first time. Sometimes there's, okay, a lot of the experiments are done at national facilities, and neutron people are used to doing that. And so you should email a beamline scientist and get help from them. Write a good proposal for the beam time, and it's a good idea to get help the first time you do that. In other words, collaborate with someone the first time, but after the first time you can do it on your own. We have a lot of data analysis software, which is all open source. Nearly all of it's open source. Some of it is Columbia owns a license, but it's still available free for academics. So there's plenty of software out there, both for data analysis and for modeling. It's all online, and you can download it, and it has tutorials. In terms of the reading, there's a couple of introductory chapters you can look at, but one of the most important things is a book that actually I co-authored with my PhD advisor, Takeshi Igami. This is a very comprehensive book. There are also a number of good review articles. So this information is on this view graph. This is front cover of the book. Okay. For neutrons, the best way to collect data is actually using a spallation neutron source, and the reason is that this has epithermal neutrons giving you short wavelengths. So reactor neutrons are less good. Spallation neutrons are good, and a very good instrument is NPDF at Lance, GEM at ISIS in the UK, and at SNS, PowerGen and Nomad. So here's a more complete list of neutron beamlines suitable for PDF. And again, contact the instrument scientists to get more information. X-rays are also very good for doing this. And the most common way of doing the experiments with X-rays is actually using the rapid acquisition PDF method that we developed about 10 years ago with Pete Chupas. And he's instrument scientist now, actually group leader at ID 11 at APS. We also have a beamline for doing these PDF experiments at Brookhaven, so you can also contact me if you're interested. These are very rapid experiments where the beam comes in, hits the sample, you collect the data on a 2D detector. So we can collect data, the record is actually 30 Hertz, so multiple data sets per second in favorable circumstances. These are the beamlines that you can do it at. You can also do it on an X-ray machine in-house, but you have to have a moly or a silver tube. We recently showed that you can also actually get reasonably reliable PDFs from electron diffraction, so that's something else you can look into. These are the software programs. So this is PDF-GetX3, which is for doing the data reduction, so going from raw data to PDFs. PDF-Fit2 is a modeling program, but it comes bundled together with PDF GUI, so this is for doing the modeling. There's a next generation software code that we're working on now that we're about to release this fall called SirFit. And this is a highly modular program, very flexible, and it's for the hardest problems around. Alternative approaches that we're not doing but are available is RMC reverse Monte Carlo, where you use a simulated annealing algorithm to get a large box of atoms to agree with the data. These are some RMC examples that come from my colleagues, Dave Keane in the UK. Another approach is called empirical potential structure refinement, which is another big box method where you have a large number of atoms in the box, but instead of moving them all independently, you constrain them with a small number of parameters. And what you do is you specify intratomic potentials, you minimize the energy of the cluster, and then you compare that to the data, and then you modify the intratomic potentials, and then you iterate this until you get a good fit. So this is like RMC, but it's actually more heavily constrained. And there's actually some very remarkable results, people mostly being done. This was Alan Soper's development, and now it's being pioneered in the UK. And for soft materials, this is probably the method of choice. The experiments are quite arduous, I should let you know. But the kind of information you can get from this is you can have a solvated molecule and you can look at the solvation structure. So these yellow things are where the solvation atoms are around this molecule at the center. And so this is my last slide. This is some community activity that if you're interested in getting into this, I encourage you to go to our website, diffpi.org. And there's a Google group where there's a discussion, fairly active discussion, tips and tricks and how to get started. So that's pretty much it. So I'm ready to take questions or have any. Okay, so Philippe has asked a question. Going back to the packing of the beta and gamma form of the drug molecule, is it possible to determine using linear combination of PDF, what is the composition of beta and gamma? Yeah, so the answer is actually yes, we can do that. We have an example that we just submitted to, I can't remember which journal actually, but one of the pharmaceutical journals where we did exactly that. And so this is very interesting. The very interesting thing that we got from that carbamazepine example was that the polymorph that we melt quenched was the gamma form, which is the most stable form of the material. when it recrystallized in the beta form, which is actually the metastable form. Presumably, if you leave it long enough, it will then transform again to the gamma form. But in general, there's going to be a mixture of beta and gamma coming out. And we've actually shown that you can do that. You can quantify how much beta and how much gamma is in the sample. And you can actually watch that as a function of time as the thing recrystallizes. So that's an excellent question. Thanks, Philippe. Are there any more questions? So I don't see anyone else typing Mayun, so actually I have to go and teach another class right now. 2 classes back to back. So Philippe says, thanks for the nice lecture, and thanks very much for attending. And let me apologize that it was at an earlier time. And that was because my conflict is that I have to actually teach my regular class at 2:40. So thank you very much, everyone. And I hope you enjoy also the rest of the course. I hope it's going well for you. Okay, thanks. Goodbye.